Welcome friends and my dear students in the Sanjeevanee Mahavidyalaya Chapoli's YouTube channel video class BSCTY. We are going to study the paper number 12, the genetic materials, evidences for DNA as a genetic material. Yesterday already we studied the Griffith experiment. And in the Griffith experiment, they proved that DNA is responsible for transformation into one generation to another generations. Today, we are going to see the another experiment. The experiment is called as Avery, McLeod and McCarthy experiment. These are the three scientists. They unitedly proved that they carried the experiment on the transformation of DNA, and they discovered that DNA is the substance which is responsible for transformations. Transformation means the genetic material DNA which transferred from one bacteria to another bacteria. There is no any other component. They proved with the help of their experiment, they proved that no any other substance which is responsible for transformation. And whatever their record, they published their record in the paper format in the 1944 in the Journal of Experimental Medicines. They published their work. Once the work is published in the specific journal, it means that the credit of that work is only for those peoples who discovered. So in this experiment they used the pneumococci bacteria. They use pneumococci bacteria. It means that pneumococci bacteria is the bacteria which causes pneumonia. And first time the pneumococci bacteria collected in the test tubes and heated on the burner. So due to which what happens? The soluble components are separated in the saline. Protein was precipitated out using chloroform. It means that different solvent systems they are used for separation of different components. For example, chloroforms they are used for separation of proteins. Polysaccharides they are used for separation. Polysaccharides are separated by using enzymes. And rather the fibrous material present in the tube, that is precipitated with the help of alcohols, and the fibrous materials finally are chemically analyzed, and it showed that there is the presence of hydrogen, presence of nitrogen and phosphorus and carbon, and these are the components similar to chemical composition of DNA. And with the help of this component and this combination, he proved that this component is a DNA, and DNA which is responsible for transformation process. So the experiment, in this experiment, apart from any other rather than the DNA, means proteins, RNA or other cell components, they are not responsible, because he used what? They used trypsin, chymotrypsin, or any other ribonucleases, and with the help of these enzymes he proved these are the enzymes. They destroyed RNA proteins, and once they are destroyed, there is the transformation. If due to the destroying protein, due to the destroying RNA, there is a transformation process happen, it means DNA is responsible. And after that, when he used the another enzymes, that is the DNase, that enzyme is also called as deoxyribonuclease. During that, DNA is destroyed, and once the DNA is destroyed, it means that there is no transformation process. And if DNA absent, no transformation, it means that DNA is the substance which is only responsible for transformation process. So this is the experiment the scientists, they perform such experiment with the help of the test tube and their materials. Basically, the requirement of the experiment is test tube, solvents, and S cells. S cells means it is the strain of Streptococci pneumonia. S cells. S cells means pathogenic strain, and the pathogenic strain he heated on the burners. Due to the heating, whatever the presence of capsule, it is removed, and once the capsule is removed, whatever the substances present inside the bacteria, for example carbohydrates, for example proteins, DNA, RNA, they comes out means they precipitated in the test tube, in the solvent. After that, the mixture solvent he distributed into the four tubes. You just see here in the test tube, the green color, it looks like in the test tube, it means there is the presence of proteins. Blue color, it indicates that there is the presence of DNA. Red color, it indicates that there is the presence of RNA. These are the components present in the bacterial cells. So let us see one by one what they perform their experiment in the first tube. This is the first tube, control tube. Why they are called as control tube? Because there is no addition of any enzymes. Remains proteins, DNA, RNA are present. Within that, there is the addition of R cell. R cell added. R cells means it is the Streptococci pneumonia non-pathogenic strain. Due to the addition, what happens? Yes, cells further present. It means that whatever the DNA, RNA or protein, any one component is transferred into the R cell, or R cell component transferred to the S cells, and there is the some transformation occurred, due to which S cells present, it therefore transformation occurred. This is the control tube. In the second stage, what happens? In the second tube, he added protease enzymes, and due to the addition of protease enzymes, there is the degradations of proteins, means protein is destroyed. Only two components remains, DNA and RNA are still present. Then within that, there is the addition of R strain, R cells of Streptococci pneumonia. So due to the addition, after a period of time, there is also transformation, means S cells are present. It therefore transformation occurred. It means that any one compound, that is RNA or DNA, is responsible for transformation, means it is one of the genetic materials which is responsible for transformation from one bacteria to another bacteria. In the third experiment, he used another enzymes, ribonuclease. Due to the use of ribonuclease, what happens? Ribonuclease is the enzymes which degrade RNA. Due to the presence, due to degradation of RNA, or due to the removal of RNA, only two components are present, proteins and DNA. And within that, there is the addition of R cells. R cells means the strain of R microorganisms of Streptococci pneumonia. Again, what? S cells present. S cells means pathogenic strains. It means that there is one is the DNA or protein which is responsible for transformation, the genetic materials. That's why transformation occurred. And in the last test tube, what happens? There is the addition of deoxyribonuclease enzymes. Due to the addition, there is a degradation of DNA, and once the DNA is degraded, separated, only proteins and RNA are present. Within that, there is the addition of what? R cells, and after that, what happens? S cells absent. Therefore, transformation are not occurred, did not occur. It means that in the in the mixture, if DNA absent, there is no transformation. Only if once the DNA absent, there is no transformation. It means that DNA is only component which is responsible for transformation, means it is a hereditary character, it is the genetic material which is transferred from one generation to another generation. And this is the experimentally the scientist Avery they proved that yes, with the help of this experiment, only DNA is a genetic material which is responsible for transformations in the bacteria. Which bacteria? Streptococcus pneumonia. So this is the experiment. With the help of experiment, we studied which experiment? A very MacLeod and McCarthy experiment. On the base, this experiment is based on evidence of the DNA as a genetic material. So student and my dear friends, so I think with the help of this video you are getting the knowledge about this paper, and if you have any problems about that, I am always ready for answering your questions. So thank you.